Hi, I'm Martin Walsh from Diamond Light Source, and I'm going to summarize the structural approach we've taken to develop antivirals targeting the main protease of SARS-CoV-2. First, I'd like to thank the organizers for inviting me to this one-day virtual meeting on SARS-CoV-2, and hopefully we can all come back in person in a year's time with further success stories to recount to each other. Structural biology is well placed to respond to a pandemic like SARS-CoV-2. So what I show here in the top right hand corner is an aerial photograph of the Shanghai synchrotron facility, and Chinese-based structural biology groups were able to exploit the fantastic MX beamlines at this Shanghai synchrotron to rapidly determine a number of SARS-CoV-2 protein crystal structures. In the bottom right hand corner is an image of a high-end cryo-electron microscope. And some of you may be aware that at Diamond we've integrated cryo-EM into the Diamond facilities, and this has led to what some people have called the democratization of cryo-EM, because up until recently accessing these high-end microscopes was really quite difficult. So we've set up a national facility at Diamond, and people can actually apply through a peer review process to access the microscopes in a similar way that they access the beamlines at the synchrotron. Now, during the lockdown in the UK, Diamond continued to operate, and we also had a special call to accommodate users with COVID-19 related research. And going back to our Chinese colleagues, so the first structure from China was that of the main protease, which was determined by the groups of Hao Tao Yang and Zi Rao. And they contacted us around the end of January to see if we could actually provide them with some beam time at Diamond, because the Shanghai synchrotron was going down into a long shutdown period. But they were also very interested to see if we would be interested in initiating an X-ray fragment-based screening experiment targeting the main protease of SARS-CoV-2. Fragment-based drug discovery takes a different approach to the traditional high-throughput screening methods, where typically millions of complex compounds are screened to find drug-sized starting points for drug discovery. Whereas with the fragment screening approach, you typically screen around 1,000 small compounds, or fragments as they're called, and the use of these less complex molecules allow us to efficiently probe the chemical space of the target of interest. And from the screening experiment then itself, we can identify initial starting points for drug discovery. Now, these fragment hits, as they're called, typically bind very weakly to the target, and we need to determine the crystal structures of the hit to be able to optimize these initial fragments into more potent inhibitors that we can then take forward to the clinic. So typically, determining the structure of hundreds or up to thousands of crystal structures is a big task, and so only recently has the use of X-ray fragment-based drug discovery become more common due to the availability of high-throughput synchrotron MX beamlines, which have a high level of automation. Now, in particular at Diamond, Frank von Delft's group have established a dedicated high-throughput platform called XChem. So this is essentially taking the whole process and bringing it into Diamond, and allowing people to readily access this technology to be able to easily perform a fragment campaign. So this just summarizes the XChem facility at Diamond. It's accessible through a peer review process. It provides the users with a complete workflow for doing an X-ray fragment screening experiment, from preparation of the samples to the collection of the data, through the whole analysis, and also the deposition of the data within the PDB. It's been up and running since 2016, and typically now a user group will be able to come and collect and process around 1,000 crystal structures within a 2-day period at the facility. So, following on from our discussions with our Chinese colleagues, we decided in the end to go ahead and carry out an X-ray fragment screen targeting the main protease of SARS-CoV-2. So just to briefly recap, the main protease is an attractive target for drug discovery, as it's essential for the replication of the virus. And so just show a schematic here of the viral life cycle. So once the virus enters the cell, The replicase gene is translated. This gene encodes for two open reading frames which express two large polyproteins termed PP1A and PP1AB, and these are made up of the viral non-structural proteins. These polyproteins then are subsequently cleaved into the individual non-structural proteins. The main protease, or NSP5, is so called because it cleaves at 11 sites, releasing 13 of the 16 non-structural proteins, while another protease, the papain-like protease, or PLPro, cleaves the remaining 3 non-structural proteins. So if we can inhibit the main protease, then we can also inhibit this proteolysis of the polyproteins chains PP1A and PP1AB, and then essentially inhibit the replication of the virus and prevent it from going on and infecting other cells and causing COVID-19. So much work has gone into the development of protease inhibitors in the case of SARS-CoV-1, but to date no drugs have been developed, and of course there's a massive initiative now to try and also obtain reasonable leads for SARS-CoV-2. So our aim was, by carrying out an X-ray fragment screen of the main protease, we would be able to accelerate the development of new leads to take to clinic. So how did we get on? This just summarizes the kind of key points from going from gene to structure. We had to actually redo everything from scratch because at the time, at the end of January, China was already in lockdown, so our Chinese colleagues weren't able to ship anything to us, so we recloned the main protease. By the middle of February, we'd managed to get crystals of the main protease, and this was a key moment because we knew from our Chinese collaborators that they'd had difficulty in growing good quality crystals of ligand-free main protease. They had successfully solved the structure of the main protease with a covalent inhibitor N3, but we're struggling to get decent diffracting crystals of the ligand-free form. As well as carrying out a standard X-ray fragment screen, we also provided protein to Near London's lab in the Weizmann, who were able to carry out a screen using mass spec of a covalent fragment library, and the hits identified for that were then triaged, and we were able to include them in our full X-ray fragment screen, which we carried out at Diamond on the IO41 beamline at the beginning of March. So in total, we collected data from over 1,400 crystals, and these were all analyzed and resulted in us obtaining 78 fragment bound structures, which we released straight away from our websites at Diamond, so that the scientific community could download and take these results and analyze them themselves directly. So we got a large number of fragment hits from the screen. This shows that we got a very comprehensive coverage of bound hits at the active site of the protease. Due to the extent of the fragment screen we did, we also got multiple overlaps of fragment hits. So here I just show some examples of covalent and non-covalent fragment hits which are overlapping at the active site. And the idea here is that you can merge these fragment hits and generate a new fragment which potentially will be more potent. And I'll give an example of this in a second. But I just wanted to go back to the whole business of making this data available as soon as we've generated it. And one way of actually getting that message out was through social media. So once we finished the experiment, the actual fragment screening experiment at Diamond, I did a tweet just summarizing what we did and that we'd released the data on our website. And this got a kind of a viral response, and also a lot of people got in touch with us. So one tweet came from an ex-member of Alpha Lee's group in Cambridge, and Alpha has recently set up a company called Postera, which is using artificial intelligence to accelerate drug discovery. So he asked could we help, and this essentially just led to an ambitious crowdsourcing initiative, which they've called COVID-Moonshoot. And the idea here is to try to accelerate the development of SARS-CoV-2 antivirals targeting the main protease. So taking the data that we've made available immediately, they also then wanted to get people to engage with that data and and help with accelerating the design of better compounds. So the response we got was really overwhelming. So very quickly people submitted designs to try out. And to date, the best compound we have is is this one here, which is showing in an IC50 of 50 nanomolar. So it's really quite impressive where we've gone since April. So we've had over 13,000 unique design submissions to the COVID moonshot from over 370 people around the globe. Of that, we've synthesized 1,000 compounds and assayed them, and we've got in excess of 10 compounds with a sub-micromolar inhibition, and we've determined over close to 200 structures. We've got funding directly from people who've just donated, and also we've got some additional funds from grants. So within this COVID moonshot, then we've set up all the infrastructure. So we've got two activity assays. One is a fluorescence-based activity assay, which has been set up in the London lab, or near London's lab at the Weizmann. And then at Chris Cosefield's lab in Oxford, we have a rapid-fire mass spec activity assay, and these are all the data from is all released onto the web, so people can compare the data generated through the assays. So having this input from people around the globe, we've been able to develop a number of lead series from the crowdsourced designs. So this is an early example from an Oxford contributor where he looked at the structures that have been determined from the fragment screen, and this inspired him to come up with this particular compound, which was assayed and showed to have an IC50 of 23 micromolar. And again, then taking this through, we were able to very quickly go from this compound to a series of other compounds which are showing more higher potency. And of course, again, this idea of taking these different fragments and merging them together will hopefully generate a more potent inhibitor. So this summarizes, so as you can see now, the concept of the COVID moonshot has emerged. We have released all the data from the fragment screening, which provides a basis for people to submit designs. And then we're now at this stage where we're essentially trying to optimize the compounds that have been submitted to the project. And this has generated a real global effort covering all the different aspects from doing the assays to chemical synthesis to molecular modeling, etc. So an example would be Enamine, who are making a lot of the compounds for the initiative, and they're doing that at cost. So in parallel to the X-ray fragment screening and the Moonshot initiative, we've also been looking at repurposing of drugs. So in collaboration with Exentia, which is a small startup in Oxford, we've been able to obtain the Reframe library, which contains over 12,000 clinically ready compounds. This was developed by the Scripps in San Diego with funding from the Gates Foundation. A number of people have had access to this and have been using it, so there's been a number of papers published. But in our case, we've actually taken a targeted biochemical screen approach to this. So we've screened both of the SARS-CoV-2 proteases, so the main protease and the papain-like protease, and we've got a number of very interesting hits. So to date, we've identified inhibitors for both proteases. We have determined crystal structures of the main protease with 2 clinic-ready compounds, and we show that these are bound at the active site of the main protease. And in the case of the papain-like protease, we've also obtained a really high potent inhibitor of the enzyme, and for both cases we're now pursuing viral neutralization studies, and hopefully we would be in a position to, if these turn out to be successful, to start human trials. In the case of the PL-Pro inhibitor, we have data already that would allow us to go into phase 2, and with the main protease, we would be optimistic in starting phase 1 clinical trials with one of the compounds that we've identified. I can't say much more than that at the moment. So in summary, with the COVID moonshot, as you've seen, these merging of fragments is quite powerful, so we're exploring immediate merges from the series that we're identifying from the contributors from the COVID moonshot. In addition to that, we're accelerating the expansion of the multiple lead series using free energy perturbation calculations, and this is again exploiting crowdsourcing. So in this case, unused CPU by the general public is being used by Folding at Home to do these calculations. The Moonshot project is also trying to get cell and live virus assays set up to achieve this goal of trying to get a preclinical candidate within the next 6 months. And as I said already, with the drug repurposing, it's full steam ahead to clinically evaluate the 3 compounds we've identified from the reframe screen against the main protease and the PL probe. This has been a massive collaborative effort. In particular, I'd just like to thank members of my group and Frank von Delft's group, which are at Diamond and shown here in red, and all the others here that I've listed. And thanks very much for your attention.